to the next, all right? That's the only way for us to get through 14 in an hour and a half, all right? So please stick to your time, all right? And we are going to mostly follow the order of the book, but we've had some requests to move some things around. So to get us started, we will welcome Jimmy Payne. Okay, go ahead, Jenny.
Okay. Just gonna make sure I can see here. Okay. So good afternoon. My name is Jenny, and I am a member of the Medical Photonics Lab under Dr. Jessica Romello-Roman. Today I will be telling you about imaging cervical remodeling in pregnancy through polarization techniques. Okay. So first, I actually would like to pose a quick, simple question to the audience. What is the reason for a hospital to have a NICU or neonatal intensive care unit? Does anybody want to take a guess? I missed that, but I'm assuming you said something about babies. Correct. It's actually very specifically preterm birth. So there is such a need for this that there's a whole unit in the hospital dedicated to it. It's the #1 cause of death worldwide for infants. And in the time it took for us to go through this, a child has died from the complications due to preterm birth. So that's why research into this field is so important. So my research focuses on the cervix, which is the organ in the body that keeps the baby in place during gestation. And it's organized in a circumferential manner. The collagen in the cervix is organized in a circumferential manner in the extracellular matrix. Collagen is the protein that actually gives our cervix its structure and its mechanical properties via its organization. And so when that changes throughout gestation, because the cervix has to go from a strong structure that keeps the baby in place to then a weak and dilatable structure that can then facilitate the delivery of the child. If this happens too quickly via collagen disorganization in the extracellular matrix, that is what results in preterm birth. So amazingly, there's actually no diagnostic tool available that actually can reliably detect a woman's risk of going into preterm labor. But as I have mentioned, collagen has a very strong role in this, and it has a property that kind of allows it to change the way light travels based on the orientation of the fiber itself. So that property is called birefringence. And with Mueller matrix imaging, which is polarization imaging, we are actually able to see that collagen fiber very well and see its organization. So Mueller matrix polarization imaging is kind of like putting on a pair of sunglasses. It kind of lets you see certain things more clearly. And with our microscope, we're able to do that in a much more accurate and precise way. So it is the perfect methodology for a diagnostic approach for preterm labor. So to validate this methodology, we start with a mouse model, because of course we're not going to go and image pregnant women. And so to start with, the reason why we chose the mouse is because the extracellular matrix where the collagen is located in the cervix of the mouse is conserved between the human and the mouse based on transcriptomic and proteomic data. The behavior of the cervix in the mouse is similar to that of humans. It keeps the baby in place during gestation. The cervix remodels similarly in the mouse as the human, as it goes from ripening to softening and dilation. And the mouse has a pressure fluid-filled cavity that then allows for the natural alignment of the collagen fibers that we're looking at. And finally, most importantly, is time. The mouse's gestation time is only 19 to 21 days, so we can study this whole entire process in a relatively short time period. So this is what we're looking at. This image is taken by a very powerful microscope, and we can see here clearly that the tunnel where the baby goes through is surrounded by organized collagen fibers that are densely packed and organized. This is what allows for the baby to stay in the womb. But later in gestation, this organization is completely lost. And if we can do statistics on this, it directly correlates to how soft the cervix is at that time. So of course, that system was imaged with a very high resolution and powerful microscope. And so that microscope has a long acquisition time and a very powerful laser. It makes it not very easy to translate into a clinical setting. So our lab has then made a simple polarization setup on benchtop so to validate our system with mouse models. And as you can see, we're able to get similar information on this day 0 sample of the cervix. And so with that information, we're also able to look at the linear retardance, which is the phase delay between the rays, get an orientation map similar to that of the high resolution image, and also look at its orientation and organization. So the orientation is kind of the different changes of color around us. So with variance and kurtosis and entropy, the circular statistics that we do on this image, we can then actually assess a woman's risk of going into preterm labor because we know how soft her cervix would be. So our lab has actually now moved on to a clinical application. We've created a portable imaging system that is using Mueller matrix. Basically, we took what I just showed you and miniaturized it into a visual system for women in clinics and hopefully maybe even in telehealth. And so how we envision this to happen is women right now, when you're pregnant, you actually go to regular checkups, right? With a physician, OBGYN. And so during this time, we believe that our device can be an accessory for the OBGYN, and they then can identify, early on, if you're at risk of giving birth too early. And this early detection is what is going to allow them to then intervene with the proper interventions medically that then can allow a woman to give birth during, with a normal gestation and have a healthy baby. So my research now is on performing a clinical trial so that we can optimize it for a diverse population of women.
Question? Slide position.
Thank you for my lab mates, professor collaborators, and funding sources. Yes. Sorry.
Sorry, I liked your presentation, very cool, very cool. I was wondering, in the ECM, obviously there's a lot of other proteins and stuff aside from collagen. So like, is there a way that you like filter for just the collagen, or do you look, like
So
how do
we
you get
actually
like back that
validate
out, I
it
guess?
with second harmonic generation, which is sensitive specifically to collagen. And then we're able to kind of see that we get similar information because polarization is sensitive to the actual fibers that collagen has. And so it doesn't actually give us as much information on other aspects of the ECM.
Okay, cool, thank you.
Okay, we will proceed on, stay in the photonics world, and have a presidential Ajmal.
Good afternoon, everyone. My name is Ajmal. I'm another PhD student from Medical Photonics Laboratory, working with Dr. Jessica Romero on stage. Today, Jenny already talked about how polarization is very sensitive to tissue microstructure, and especially tissues with collagen, a lot of collagen in it. And in this presentation, I will show you how, the issue is that when the sample is biologically thick, a lot of this polarization information gets scrambled as the thickness of the sample increases. And in this presentation, I will show you how we developed a polarization sensitive confocal imaging microscope which does optical sectioning, and how we recovered polarization information from thick biological tissues at different depths. So, Jenny already mentioned all these things, that polarization microscopy has got immense interest in biomedicine as a label-free, non-invasive technique which is highly sensitive to tissue microstructure. These are particularly sensitive to optical anisotropy, which makes this technique very useful for visualizing fibrous tissues like collagen and muscle fibers, nerve tissue. But the issue is that conventional myelomatrix imaging, like many other imaging modalities, will give us a 2D projection of the sample. For example, and then what happens is that when it gives us the 2D projection, it does not distinguish between the origin and nature of these polarization parameters. that arises whether from a surface or subsurface layers of this tissue. For example, this is a mouse cervical tissue image obtained from a polarization imaging microscope, and these are specific information that we can extract using our polarization technique. For example, this is a linear retardance which shows the optical anisotropy within the tissue, and as Jenny mentioned before, we see a higher linear retardance value which indicates that the presence of densely packed collagen fiber within the cervical stroma of the tissue. Now the question is, where did this linear retardance value arise from? The thing is, with the normal millimetric polarization imaging microscope, we can definitely extract polarization information, but we cannot reliably assign a specific depth to its origin. So to address this issue, we developed a multimodal imaging system that consists of a back reflectance millimetric polarization imaging methodology, and it has a polarization sensitive confocal imaging polarimetry which does the optical sectioning, also gives us polarization information from different depths, and which is validated with a co-registered nonlinear imaging microscopic system that would give us nonlinear signals like SHG, second harmonic generation imaging, and two-photon fluorescence imaging, where SHG is specifically considered as a ground truth information that gives us spatial orientation or like spatial distribution of collagen fibers. So here is a schematic of our system that uses a femtosecond high power laser. We use different optics parts for the generation of polarization states of light and for the analysis of polarization state. And this, as I said, this multimodal imaging system would give us nonlinear signals, SHG, and quantitative polarization data. And the nonlinear signals complement the presence. It complements the information, and it gives us a very specific information about the distribution of structures like collagen fibers. Here is a result from our microscope that shows the nonlinear signal from a mouse cervical tissue. The leftmost shows the SHG and two-photon signal from a cervical tissue. As I said before, SHG signal indicates the presence of collagen fibers, and as we go to the right, the M11 is like a intensity image collected from the sample, and the linear retardance is the optical anisotropy map, which which you can see that it matches with the collagen distribution that is being depicted by SHG signal there. And on top of that, we can also get the orientation information of these collagen fibers through this technique. While this shows the normal, regular two-dimensional images, we also were able to get the information. This is like SHG and two-photon signal from the sample, and here is the confocal image, that confocal millimetric images that we get. What we can do is that we get a digital mask out of the nonlinear signals that would specifically tell us the presence of collagen fibers in the tissues, and then we apply that onto the polarization information that we extracted from the sample, which makes sure that the polarization information are selectively and specifically coming from collagen fibers. And these are depth resolved polarization information that we extracted from the sample. And not only we qualitatively represent them, we also quantitatively extracted those information, and this shows that we are now able to extract polarization information from thick biological tissues at specific depths using our polarization sensitive confocal imaging system. And with that, our conclusion is that we were able to make build a multimodal imaging microscope that has co-registered nonlinear as well as back reflected millimetrics as well as depth resolved millimetrics imaging. And we were successfully able to extract this polarization information from thick biological tissues at specific depth. And with that, thank all my
Questions for Ajmal?
Really good presentation.
Thank you.
So you said the magnification that you have right now is 5.
This is a 5x magnification.
Yeah. Do you
Yeah.
think you could get higher?
Yes, we can go higher. We are working on that. We'll switch it into high magnification. But this one, we just wanted to show that as a collective information from the entire cervix. But as we go into higher magnification, we'll have to stitch them together and combine it together.
I'm definitely asking for myself. We'll
Thank
chat
you.
later.
Thank you so
Thank
much.
you.
Next up, we have a presentation from Sean Moore.
Good afternoon, everyone. Today I'm going to present part of my dissertation work, which is precision medicine for thyroid cancer. So in this aim, we're gonna use the pharmacokinetic model trying to optimize the current IDA-124 imaging protocol. So all right, so thyroid gland is part of our endocrine system, and it uses the iodine to produce a hormone that helps regulate our body metabolism. So for the thyroid cancer, actually, most retain this iodine uptake capability, so which allow us to use radioactive iodine for imaging and treatment purpose. So after the surgery, radioactive iodine is used to ablate the leftover tissue, they call remnants, and also any nodal and distal metastases. And the radiation dose is very critical for radioactive iodine treatment. So in current recommendation is basically we calculate the cumulative activity over the region of interest and times the energy deposition kernel and divided by the mass that give us the mean absorbed dose. So in that case, we need to have a time activity curve, and the total cumulative activity will be the area under the curve. So current imaging protocol it requires 5 days a whole week. So basically Monday to Friday. You can see it's a great burden for the imaging center and the patients. So we all know like a simple two compartment model actually can be proposed for the thyroid and extrathyroid pool. So by using the differential equation to describe the thyroid exchange, we can solve the activity inside the thyroid with by exponential solution. Actually, this is a typo here. It's actually 3 parameters. So basically, A is the scaling factor, lambda 1 is the clearance constant, and lambda 2 is the absorption constant. And the Tmax actually can be calculated based on the other parameters. So our aim is, to use the PK model to calculate the accumulated activity and also trying to reduce the number of imaging points while maintaining the accuracy. So this is a phase 2 clinical trial. So 6 patients enrolled and 13 remnants, 13 lesions are identified on the PET image. So what we're doing is we use a 5 days imaging scan and use MATLAB and R to do the model feeding for this by exponential solution, and use this 5 days plus also time 0, the activity 0, use it as a reference. So to calculate the accumulated activity, and then by reducing one individual imaging time point at first, we do the same analysis and compare the 5 days reference, and then later on we further reduce more combination of the imaging points. So this is the first is removing one image day. We can see that early phase like 48 hour and 2 hour are very important. That will stabilize the absorption part, and the later part, actually 72 and 96 hour, is kind of redundant from each other, because as long as one of them actually is capable to capture the clearance phase. And then we further reduce to like 3-day points. So we can see actually those without a late measurement actually has a larger bias and variation. So that means a late measurement point is very crucial for accurate constrain the clearance phase. Then we further reduce to do 2 points only, and the 2 points plus the initial 0-0 condition. That's the minimum you need to solve this 3 parameters equation. So the 24 plus 96 hours that give us a very promising result. It's kind of makes sense because 24 hours kind of give you the uptake phase, and 96 hours is the requirement for the clearance phase. So then we bring all this combination together. You can see those are the two best combination from all these different imaging options. So further move forward, so is it possible to do a single point, right? Because there's a lot of research doing this. So the easiest way to single point is just use the patient's population average data. So we did a cohort average parameter, and then we can see that the early imaging points actually give you a larger bias and variation, and later phase give you a more robust dose estimation. So future work will involve more larger data sets and the tumor-specific kinetics, and especially with the absorption and clearance parameter actually has underlying physiological meaning behind it. So this may further refine our single or 2 points dosimetry, and which advance our personalized radioactive iodine treatment. Thank you.
Question for Sean? Next up, we have Anna Valentine.
Hi everyone, my name is Anna Valentine. I work with Josh. And today I'll be talking about the effect of relaxin receptor agonists on medial versus intimal vascular calcification. Okay, so vascular calcification is mineral that is deposited within our arteries. And when this happens, our vascular smooth muscle cells undergo stress. And this stress causes them to osteogenically differentiate. So they become bone-like cells. And they start secreting this calcification mineral. And right now, vascular calcification is one of the best predictors of cardiovascular morbidity and mortality. There's two types of vascular calcification, and I'll go through both of them today and explain why we're looking at both. So first we have medial vascular calcification. So this occurs within the vascular smooth muscle layer, so this inside layer of our arteries, and it's often associated with chronic kidney disease due to the phosphate imbalance in the blood. These patients are often at risk for heart failure. And then we have intimal vascular calcification, which is probably what you're thinking about when you think about a heart attack or a stroke. This is calcification that occurs within the lumen, so where blood is actually flowing, and it's often associated with atherosclerosis, which is that lipid deposition that occurs within our blood vessels. So unfortunately, right now, there are no therapeutic options to treat vascular calcification. And so we hypothesize that relaxin may be a good therapeutic target for this pathology because of its vasoprotective and antifibrotic properties. Unfortunately, recombinant relaxin is poorly stable, it's poorly bioavailable, and it's really expensive to synthesize. And so we work with the NIH NCATS team, the translational sciences team, to develop a small molecule allosteric agonist of the human relaxin receptor. And this is what we use, ML290, to test whether or not we can treat vascular calcification in vivo. And so today I'm going to show you our experiments on whether or not we could do treatment on both medial and intimal vascular calcification. So first we treated mice that had a chronic kidney disease in order to test for medial vascular calcification. We did this by giving them 6 weeks of a high adenine diet to induce renal failure, and then we gave them a high adenine and high phosphate diet to induce medial vascular calcification. During the last 2 weeks, we also gave them either vehicle control or 10 and 30 milligrams per kilogram of ML290. Finally, right before culling, we gave these mice a tail vein injection injection of OsteoSense, which is a probe that binds to this calcification. And it allows us to image how much calcification is in the aortas when we use a LiDAR scanner. So we quantified the mean intensity of the aortas, and we found that we were able to significantly reduce vascular calcification in these mice, particularly in the males. We also found that in the females, due to a diet-dependent problem, there was no significant calcification increase, and so we weren't able to see any effects of ML290 on the females. Then we tested it on intimal vascular calcification. Again, this is an atherosclerotic mouse model. And so we used Poloxamer 407 or P407. This is a surfactant block copolymer. So all that means is that we're able to inject it into these mice and we give them hyperlipidemia. We increase their lipids in their blood and then we cause them to have that atherosclerosis in their blood vessels. We did this injections for 25 weeks every 3 days. And then we gave them ML290 again at 10 or 30 mg per kg for the last 10 weeks of the experiment, and again injected them with OsteoSense in order to visualize the calcification within the aortas. So when we did this, we took the fraction positive, or the percentage of calcification in each of these aortas, and we found that we were able, again, to significantly reduce vascular calcification at both 10 and 30 mg per kg, especially and most significantly in male mice. So after completing all of this, we found that we were able to significantly reduce vascular calcification in both the chronic kidney disease and atherosclerotic mouse model. And we also noted that there were some sex-dependent differences in the effects of relaxin receptor agonism. So thank you to my lab and all of my funding sources.
Question for Adam.
Just a quick question, basically, what do you suspect is the reason for these sex differences?
Yeah, so one of the reasons, particularly in the CKD model, was actually the sodium imbalance in the diet. So we noticed that when we cause renal failure, if the diet has an increased amount of sodium, their mice are unable to properly filter, and so they have sex-dependent differences. The females just have they're more robust, so they have a better tolerance for kidney failure. So in terms of the CKD diet, that's why we saw it. For the Poloxamer or atherosclerotic model, that model is actually a very new model for vascular calcification, and so I think that we were just underpowered because we did see a decrease. It was just not statistically
Our next talk is by Lopez.
I love the way you put that together.
My name is Manuel Perez. I'm also with the Autism Lab, and I'd like to speak to you today about how we want to study how the stem cell extracellular vesicles or exosomes can be used to treat heart failure. And specifically, how can we upregulate the production of those vesicles by dynamic conditioning of the stem cells. Okay, so in a heart attack, you have an initial site of injury due to blockage of the oxygen. Initially, that's a defined area of cell death. It's apoptosis due to the lack of oxygen. However, this initial injury happens between 15 and 20 minutes after the infarct. After that, in a few minutes and hours, the injury keeps growing and affecting cells surrounding the initial injury site. And at after 4 to 6 hours, the amount of cell death is for about 4 to 5, 6 times higher than the initial injury. This occurs like, it's called the wave front of necrosis, where the necrosis spread surrounding the original injury site. The apoptosis happens inside the cells in the mitochondria, which is affected, and specifically the lack of oxygen deprives the electron transport chain of the last oxygen acceptor, and that's what happens is the electron chain completely is arrested, and all the ions, hydrogen ions that are produced are accumulated, and they don't have a place to go back into the mitochondria. What happens is, at that point, if you reintroduce flow to the heart, to those heart muscles and cells, the electron transport chain is reactivated, and more reactive oxygen, hydrogen ions are produced, and that causes an accumulation, actually, of excess hydrogen ions. And that produces oxidative stress to the mitochondria, causing the cell death. So in other words, what happens is you have an original injury due to the initial heart attack, and then you have a secondary, additional injury when you reintroduce flow back to the system. So up to now, when you have a heart attack, the treatment you have is just to get reestablished flow to the heart. But apart from that, there's nothing that can be done. So some researchers have been studying to try to get stem cells injected into the site of injury to see if they engraft and regenerate new heart tissue, contractile, so that replace the dead cells. However, it's been found that in the site of injury, it's a hostile environment. It's low oxygen, high acidic. It's not conducive for the cells to engraft or grow. So most of those cells, within minutes or hours after the transplantation, they just don't remain. However, there have been shown that about 5% of these cases, they do see some benefit. In other words, the conservation of ejection fraction of the heart. So there's some benefit that has been shown. What they found is that the stem cells themselves are not what's becoming new tissue, but they leave behind secretions that themselves are the ones that are having the beneficial effect. We want to study specifically the extracellular vesicles that are produced from the stem cells, and those vesicles recapitulate or have the same benefits of the parent cell. In a vesicle that's non-living, they don't die, so they're not affected by the low oxygen. So we want to study if these extracellular vesicles can be used to treat heart injury. This is a electron micrograph of EV that was obtained in our lab. It's about 160 nanometer in diameter. And those are the ones we plan to use to treat hypoxic cardiomyocytes. To stimulate the production of these EVs or vesicles, what we want to study is to have the stem cells produce or stressed by shear flow to see if they react in a defensive manner and start producing excess West vesicles. We have a pump in our system where we see the cells and introduce them into two different kinds of pulsatile flows. After 3 days, we collect the media and isolate out the vesicles. We have so far run a N-of-1 experiment. This is the data we have. Upper left is a control group, no flow, no shear stress. That's the size distribution of the vesicles. In the middle is pulse. and the right is oscillatory. A little bit, in this case, larger size vesicles, but we have to show you that this is N of 1. What we do have is, in terms of number of particles, static is higher, produce more particles, but in the amount of protein, the oscillatory and positive produce more protein overall, and especially more protein per particle. Next step is we're going to place cardiomyocytes in an aerobic chamber to recreate hypoxia, and then at that point we will treat the hypoxic cardiomyocytes with EVs and test the cardiomyocytes for viability after hypoxia. With that, I'd like to thank my lab. Everyone in there in the list has helped me. That's it. Okay, so any questions? Thank you.
All right, we're gonna try something here, see how this works. So Madhuri, can you, you should have sharing access, so if you'd like, you can share yourself. We have one student who is up in Virginia with a faculty who recently relocated, and he is going to try to share.
Okay.
See if you can share your
Can
screen.
you,
Or
can you hear me?
we can hear you. Do you want to do it through my computer, because I gave you access, so can you load your slides here?
Okay. I have the access, but I cannot open it right now.
Hold on, I'll do it for you. Give me a second, I'll open the.
Okay, sure, yeah, sure, thank you.
Are you able to transition? Perfect.
I think yes, I can do it, yeah.
All right, thank you, go ahead.
Thank you, Dr. Hassan, for giving me the opportunity to present over Zoom. Hello everyone. Today I'm going to present my overview of my work, PhD work. The title of my work is the development on validation of tissue curvature correction models during near-infrared spectroscopy imaging toward diabetic foot ulcer assessment. I am working with Dr. Anuradha Godavarti and I have recently moved to Virginia Commonwealth University. Let's read about diabetic foot ulcers. There are 589 million people all over the world affected by diabetes in 2024, and this number is expected to rise to 853 million in 2050. 1 in 3 people with diabetes affected by diabetic foot ulcers in their lifetime, and 20% of diabetic foot ulcers results in lower limb amputation. The gold standard treatment or assessment of diabetic foot ulcers is the color assessment, Malgardot or size, which is all of them are subjective assessment. Near-infrared spectroscopic imaging can provide the objective assessment of diabetic foot ulcers in terms of tissue oxygenation measurements. In our lab, optical imaging laboratory, we developed a smartphone-based oxygenation tool, that is SPOT, that uses smartphone camera as a detector. It has multi-wavelength LEDs with 690, 810, and 830 nanometer wavelength. It also has a 645 nanometer long pass filter to filter out the ambient light. It uses an Android application to control the device. The device was validated against a commercial device, and now we are using this device for different kind of clinical applications, such as effect of debridement and kind of healing, assess the healing status of diabetic foot ulcer. However, when we take the images in the clinic, we realize that the foot and wound has different kind of irregular and curved geometry. And when we are taking the images, there is a effect of height. There is a gap between the imaging plane to the imaging surface of the tissue. And also there is an angle between the imaging plane to the surface normal of the imaging surface. So we need to correct the effect of depth from the 3D surface to the 2D imaging plane. And also we need to correct the effect of angle between the surface normal to the imaging direction. Hence, the objective of my research is to develop and validate curvature correction models for our near-infrared spectroscopy imaging device. So I break down my study into two two studies. First one is how to develop the correction models. In Monte Carlo simulation, I developed 4 correction models. First one is height correction. Second one is Lambertian correction, that is also called angle correction. Third one is both height and angle correction, and fourth one is the empirical correction. Because of the limited time, I am not explaining all the details about the correction models, but the application and the results of the correction models. So I created one mimicking irregular geometries in Monte Carlo simulation with 3-layer tissue properties: epidermis, dermis, and subcutis. The top one is our foot model. I was trying to mimic this foot model with the wound. Then I collected the diffuse reflected signal. 2D, first one is the 2D diffuse reflected signal of flat surface, curved surface, and corrected signal with each correction models. The second row shows the profile plot, and the third row shows the percentage error before and after correction. After comparing the percentage median error, we could see that the model 4, that is our empirical correction, that could reduce the error significantly from 13 to around 14% to 1 to 2%. So, we concluded that empirical correction corrects the signal closely to the flat surface signal and provides the minimum percentage error. So after developing the correction model and validating in simulation, now we have to validate in phantom study before going to the clinic. So how do we validate the phantom? First, we have to create the phantom. So we created a fabricated PDMS-based resin phantom with titanium oxide and India ink absorber and a scatterer. After mixing, we poured them in the curved phantom mold and remove the bubbles, and then we created 7 tissue mimicking phantoms with different kind of geometry: convex curve, flat phantom, convex curve, and also foot model phantoms, the similar as of simulation. Here I am only showing the results for our irregular foot mimicking phantom. This is the geometrical structure, like the geometry of the tissue mimicking phantom, and we collected the signal, diffuse reflected signal, from the flat surface and the curved surface, and then applied the correction models. Then we calculate the percentage median error, and we could see that the before correction, the empirical correction could reduce the error from 13 to 9, from around 9 to 13% to 3 to 2%. So we concluded that we can use the empirical correction model to correct the tissue curvature issues and defect. So my ongoing work is to collect the clinical data to apply the correction model, whatever I have collected from University of Miami, and now I am working on the data analysis of the curvature correction models. So in conclusion, our near-infrared spectrum imaging device requires the curvature correction, and we could develop the correction model in Monte Carlo simulation, and we also validated the correction model in phantom studies. So I thank our team from FIU and also thank our team from VCU, and thank you everyone for your attention.
Question for Madhuri? If not, we will continue to move along. Thanks, Madhuri.
Thank you so much.
Is Alexi in here?
I'm here.
Okay, good. So I'm here.
I thought I might have been next. I didn't have a vote.
Just got a vibe.
Yeah, I don't know. Well, I counted earlier and I was like about halfway. Yeah, it was really big now.
I know. A lot of work later here, apparently. You definitely win the award for the biggest power.
Yeah. I don't know why it's so
Our next presentation will be given by Alexi Switz.
So good afternoon, everyone. My name is Alexis Swetz. I work in the Prasad lab for. What do I do?
More?
Here.
More?
More.
Yes.
Oh, sorry. Okay, well, okay. So I work in Dr. Prasad's lab, and today I'm going to be talking about bio-inspired and electrospun helically structured nanofibers for cardiac patch applications. So why are we interested in investigating these fibers? And the answer to that is myocardial infarction, which is commonly known as a heart attack or an MI. So it's estimated that every 40 seconds, someone in the United States is going to experience a heart attack. This results in a mean hospitalization cost of around $30,000, with more than 620,000 hospitalizations each year. These heart attacks are especially bad because they often lead to a section of deceased cardiac tissue caused by the lack of blood and oxygen, lack of blood and subsequent lack of oxygen and nutrients to the section of cardiac tissue. Now, the cells that make up the heart are cardiomyocytes. These are the heart muscle cells. And cardiomyocytes cannot proliferate, meaning once they're dead, they're gone. They're not going to heal themselves. So a treatment option is going to be necessary. However, the only current approved treatment option is a heart transplant, and there's a really long waiting list and a high risk of immune response and rejection. So alternative treatments are really important. So this brings us to the idea of a cardiac patch. So researchers all over the world are looking into developing essentially a living tissue band-aid, so a little piece of cardiac tissue that could be adhered on to the section of damaged tissue in hopes of restoring some heart function. So there's a variety of production methods that people are using and a variety of scaffolds and a variety of cell sources to kind of make their cardiac patches. But for the purposes of my dissertation, we'll talk about my cardiac patch, in which we're doing kind of a two-section patch. The first component of it is going to be helically coiled electrospun fibers. And the important part here is the helically coiled. So think about a bunch of like slinkies kind of stretched out in a row. They're going to be pretty flexible, right? So we hypothesize that if we can create these flexible helically coiled fibers, that flexibility is going to be really beneficial to serve in a dynamic, constant beating heart. Then we're going to encase those fibers in a hydrogel that's seeded with those cardiac cells that I was talking about earlier, the cardiomyocytes. So that way we actually have a piece of functioning heart tissue. So in order to create these fibers, we designed and built an electrospinner in the lab. An electrospinner is basically just a machine that uses a high voltage electricity to create like plastic cotton candy. Our electrospinner we use to produce these three different types of fibers. And the really important ones that we're going to talk about are the coiled fibers and the aligned fibers. The random fibers people produce everywhere, so they're not super interesting to study. Yeah, we're focusing on the coiled and aligned fibers. So once we produce the fibers, we go ahead and do a bunch of characterization testing on them to actually see how they perform. The first kind of step of testing that we do is actually an image analysis testing. So we see how the fiber diameters are coming out. And since we are comparing two different fiber geometries, right, the aligned and the coiled fibers, we've tried really hard to get our fiber diameters to be about the same. Now, our fiber diameters are falling between an average range of 20 to 25 microns, which we're counting as about the same because it's virtually impossible to get them any closer. The other testing that we do and shown here is the mechanical testing or the tensile testing. We're specifically looking at how flexible the fibers are, so what's the elastic modulus. And this is actually an old figure that I have here, but the coiled fibers are much more elastic and therefore much more flexible than the aligned counterparts. So then in terms of, we have these fibers, we've characterized them, we actually have to make little heart tissues out of them. So these are some images of what our cardiac patches actually look like. Something that's cool about our cardiac patches, and talk to anyone about it more later if they're interested, is we put them on these little holders. So they're actually freestanding tissue structures, which allows the cardiomyocytes to beat on their own the way they would in the human body, not just the way they would because they're adhered onto a piece of plastic. So we created the coiled and the aligned fiber patches. And we also tried to do a group with just the hydrogels. However, they weren't strong enough, so they didn't survive. So our future groups, we only look at the coiled and aligned fibers. So this is some staining imaging shown here. They're fun videos, in which you can see the blue is the Hoist nuclei staining, and the green is the sarcomere alpha activin staining. So just quickly in conclusion, we built the electrospinning setup, we created the fibers, we did some testing on them, and we actually built our patch structures. Thank you.
Question for us?
Yes.
So do you have any reasons to believe that your cardiomyocytes will actually beat within the structures?
Yes, so we have made them, and we actually can see them beating in the structures.
Synchronously or just each of them is doing whatever it's doing?
So far it's been synchronously. I don't have any videos, so these freestanding tissue structures, right? We have a piece of PLA, so solid plastic, that's holding them. So we have to image them from the bottom using a microscope setup that I 3D printed. So we can see them beating synchronously, I just don't have any good videos of it. But if you have any imaging videos, we're happy to have lots of anyway, yeah. Oh, yeah.
Next presentation is by Claudia Ponce.
I'm going to talk about microfatty depletion and its effect on calcific aortic valve. So calcific aortic valve disease reported about 11 million cases in 2021, and it has a death toll of about 130,000 annually, which is a number that is only increasing. And the aortic valve has about three leaflets, three leaflets, Rosa spongiosum and ventricularis. And what calcific aortic valve disease is small accumulations of calcific deposits that happen, especially on the fibrosis side, where there is like a small endothelial injury that allows for infiltrations of immune cells, and then eventually resulting in babula interstitial cells changing from their original state into osteogenic lectron state. But to understand the mechanisms that happen in the heart valve, we gotta understand the type of cells that are in the aortic heart valve. And some of those cells are babula endothelial cells, interstitials, melanocytes, which we all know, but also immune cells, with one specific immune cell being resident macrophages. Resident macrophages are hematopoietically derived. They come from the fetal liver and from embryonic development. So they're always there in your heart. And just a complete overview a small overview of macrophages. They are relatively large in size. They move around a lot, so they require a lot of ATP for that. And they move around a lot because they eat a lot of vacuoles, granules, pathogens, and engulf a lot of those. And we have two types of macrophages in our bodies. We have monocyte-derived macrophages that come from the bone marrow and then eventually differentiate into macrophages. And then we have the resident macrophages that don't have the CCR2 marker. So what I want to do with this project that I'm currently working on is identify the role that these resident macrophages that are always present in the heart have on the aortic valve, on the homeostatic conditions, and then later on in calcific conditions. But before I do that, I wanted to introduce this drug that I use. You can administer to your animal model, and this drug can depletes macrophages, it accumulates in the cytosol of the macrophages, stopping the ATP production, and then eventually these macrophages are completed, depleted. So using the drug, I can then create a research design in which I can use both female and male mice and inject them for at least 6 weeks, once a week. And there's some markers that I want to check for in here. The first marker that I want to check for is if I deplete the macrophages, I don't want to see my macrophages in there. So I'm going to check for F480 marker, which is the surface marker for macrophages, and then for elastin and collagen. For some of the preliminary results that we have right now is that the mice that did receive depletion of macrophages have lower elastin and lower collagen. Of course, this is only preliminary, I have to study more. But some of the next steps are to complete the whole experiment and then continue on to calcification, in which we're going to administer the same clotting liposomes and remove macrophages, but these mice would have undergone chronic kidney disease diet, which our lab has shown to for calcification. And in conclusion, immune cells like resident macrophages are always present in the aortic valve. They play a role in homeostasis, but we're still not clear what the role is. Preliminary results by depleting the macrophages show that there was significant decrease in collagen and elastin, and future directions are to identify the role of the cells in calcification. Thank you. Questions?
Oh, we have one.
Your presentation, do you think the timing in which you're looking at the ECM is important here? Like when you think about elastin, since this is kind of stopping around P7 in these mice, do you think if you look earlier you would see differences if you depleted the macrophages?
Well, ideally, I wanted to look at a longer extracellular matrix exchanges because it only makes sense that the longer it is, the more changes you'll see in the ECM. But as you know, my mice kept dying, so I had to lower the time from 8 weeks to 6. And I don't think that in about 3 weeks I'll see the same effects as I see on 6 weeks. But that's something we should do. Thank you.
All right, next we have Kumar Saha.
Hello, good afternoon everyone. I'm Devashisha. I'm working with Dr. Sukias. So my research is about multi-scale modeling of blood flow regulation. Blood flow regulation in the brain. Actually, I am trying to investigate how the perfusion is distributed based on the arterial and capillary stimulation. The brain has an intrinsic mechanism to adjust their cerebral blood flow to match the regional energy demand, and the regulation of cerebral blood flow processes that spans from the subcellular signaling mechanism at the microscale to the modulation of vascular tone and the tissue level hemodynamics at the macro scale. And the tight regulation of cerebral blood flow is very important for normal brain function and can be disrupted in various conditions like Alzheimer's disease, stroke, vascular dementia, different types of diseases. But still, the precise mechanism that controls the cerebral blood flow is under investigation. And the regulation of cerebral blood flow especially has two mechanisms: cerebral autoregulation, where that stabilizes cerebral blood flow despite the fluctuation of blood pressure by muscle cell contractility. The another one is neurovascular coupling that modulates the cerebral blood flow in response to neuronal activity. The emerging understanding is that neuronal activity releases different mediators like potassium ion that affect the vascular tone in the penetrating arteriole to control the blood flow. But the recent evidence from our collaborator suggested that the capillaries can also sense this neuronal activity due to their close proximity to every neuron, and it initiates the receptive signal to control the blood flow. So we aim to develop a multi-scale model of blood flow simulation in a dynamically changing macrovascular network to gain insight into this mechanism of cerebral blood flow. So we reconstructed the macrovascular network from the mouse brain imaging data, and our lab also developed this same single cell level model of capillary endothelial cell and pericyte, and arterial endothelial cell and muscle cell. And our model also developed the biomechanics model that can relate this membrane potential and calcium in small cell and pericyte to the conductivity of arterioles and capillaries throughout the network. And we simulate the network hemodynamics as the diameter are dynamically changes, and we assume Higgins-Poiseuille's law in every vessel segment. And we also consider the Fourier-Lindquist effect and the plasma skimming using the empirical correlation. In the result section, so figure A and B, they predicted the flow and the pressure distribution in a macrovascular network in static condition, and we also investigated the changes of pressure, blood flow, pressure, and diameter. So due to the myogenic constriction that is shown in figure C, D, and E, and figure P summarizes the total network inflow with respect to the pressure fluctuation in different cases like the fixed diameter and the passive tone and with the myogenic active tone. And from this figure A, the green line represents that the total network inflow is constant, almost constant for a wide range of pressure. So that means this is happened due to the myogenic autoregulation. That means our model is able to capture this myogenic autoregulation. And then we also investigate the perfusion redistribution due to arteriolar and capillary stimulation. In penetrating arterial stimulation, we stimulated 100 micrometer of penetrating arteriole by 10 millimolar of potassium ion. And we see that under baseline condition, the model predicts only localized hyperpolarization. And that's why the blood flow increases a little bit. That is shown in figure B. And when we use the high KR channel activity, this hyperpolarization can spread to the upstream penetrating arteriole and increase the blood flow. And the right figure shows that we applied different level of hyperpolarizing current to the same penetrating arteriole. And we see that the perfusion for the stimulated penetrating arteriole, here the penetrating arteriole 1, the blue line, after when we apply high hyperpolarizing current, it plateaus. That means doesn't increase so much. Whereas in the neighboring penetrating arteriole, blood flow increases. So that means this possibly inefficient distribution may either reflect a protective mechanism or suggest the existence of an optimal stimulus level that maximizes the perfusion otherwise. And for capillary stimulation, we see the similar type of things. For local stimulation, local hyperpolarization, and when high KR activity, that spreads to the upstream arteriole and blood flow increases. And the bar graph show that in layer 6, we have 30% of blood increases in high KR activity compared to only 6% in local stimulation. And that also represents that there is a conductive mechanism. And that's why this signal can spread into upstream arteriole and increase the overall blood flow stimulation. So the conclusion is we develop a multi-scale modeling approach to gain insight the mechanism of cerebral blood flow. That's all. Thank you.
Question or no? No? All right. Thank you again. So next, we will continue. Dr. Sudi gets the lab and presentation. That will be below high work.
Hello everyone. Today I will present our research on electromechanical coupling in cerebral arterial macrocirculation with a focus on the role of capillary peristaltic in blood flow regulation. To start, let's take a closer look at the brain vascular architecture. At the surface, we have parallel arteries which supply blood into the cortex. This gives rise to parenchymal arterioles serve as a critical bottleneck that regulate blood flow into the deeper brain region. And finally, blood is distributed into the capillaries where pericytes are located. On the left side, we have experimental data shows that arterioles respond to pressure. As pressure increases, arteriole diameter decreases. Also, recent evidence suggests that capillary pericyte in the first few generations may respond similarly. But the big question is that does that actually matter for blood flow regulation? Or in other words, can a micrometer level constriction in a small part of capillary network affect the cerebral blood flow regulation? That's the central motivation for our mathematical modeling. To investigate how cerebral blood flow is regulated, we developed beta cell level model electrophysiology and calcium dynamics. We developed arterial endothelial cells, smooth muscle cell, capillary endothelial cells, and capillary pericyte. And by coupling them together, we create a multicellular models of arterioles and capillaries. So we understand how cerebral blood flow is regulated. We need to link electrical activity with mechanical response. So we introduced a minimal biomechanics model that translates pericyte, smooth muscle cell, calcium, and membrane potential to vessel diameter. The model accounts for passive or elastic properties of vascular wall as well as for active zone. And the model, the degree of activation is assumed to have a sigmoidal relationship with the calcium and the Laplace can relate total wall stress, wall thickness, and transfer pressure to vessel diameter. First, we examine the response of parenchymal artery smooth muscle cell to increase intralaminar pressure. As pressure increases, some mechanosensitive ion channels open, so like NSC channels open. So in that case, we have a membrane potential depolarization. Voltage operated calcium channels open, calcium comes in, we have calcium elevation. So in that case, vessel constrict. On the other hand, under passive condition, when calcium is removed, vessel fail to constrict and instead dilate progressively, as you can see in the dashboard line. So the model capture experimental data and capture the key mechanism underlying the migraine response. And then we apply the same framework for the capillary pericyte. As pressure increases, we have a membrane depolarization, calcium elevation, and vessel diameter decreases. So these findings support that pericytes are not just passive structural cells. They appear capable of active and pressure-sensitive myogenic response, similar to smooth muscle cell, but with distinct functional role in capillary pericyte regulation. And to make our model more accessible and user-friendly, we developed graphical user interface for neural cells. These tools allow user to simulate electrophysiology and calcium dynamics. So for example, here I'm doing pressure ramp simulation for capillary pericyte. You can easily change the pressure values or switch to passive simulation by removing the calcium. And also you can do the simulation for the agonist under parenchymal arterial smooth muscle cell. The next panel shows all channels conductance. We have all NSC channels, and you can easily plot transmembrane currents, pumps, and exchanger. And finally, in collaboration with the Devasish, we extended this work into a full vascular network with 50,000 vessel segments, and cellular dynamics are simulated at each vascular network, each vascular segment to predict electrical and calcium signal. Panel A and B shows the flow and pressure simulation, and panel C shows the diameter changes. As pressure increases, the constriction extending from arterioles to the first regeneration of the capillaries. And panel D summarizes what happened to the total blood flow regulation. For example, in the blue line, you can see that when both arterioles and capillaries constrict, we have the most stable perfusion along the larger
to provide people with movement impairments control-assisted robots by harnessing really any high-dimensional bodily signals that they can still reliably modulate. Signals like, whoops, the electrical activity of their neurons, their muscles, or the kinematics of their body motions are all high-dimensional enough in theory to control assisted robots. But in practice, it's really difficult for people to learn to control robots using these kinds of abstract signals. So in my research, I look for ways of making this kind of control easier. And for my investigations, I use a model system where a healthy person controls an assisted robot using the 19 joinings of their fingers. So using the model, I can test many people and ask really general questions about how we can make the control easier. On the right-hand side here is a flow diagram of our model. So we take the user's finger joint angles, we pass it through some user-to-robot mapping algorithm, and it spits out a command U of t for the robot. In the literature, U of t virtually always represents a command for the velocity of the robot's gripper. But in theory, it could represent any aspect of the robot's motion, including the angles of its revolute joints, the speed at which those joints are rotating, or even the position and orientation of the gripper directly. The possibilities are called control nodes, and the field's focus on gripper velocity control has left open the possibility that the control would be easier if we just used a different control node. So to investigate this possibility, I brought a group of healthy people into the lab, and I provided them control of an assisted robot using our model, and asked them to come by on 4 different days. Each day, they piloted the robot using a different control node. And when they came in, I asked them to use the robot to pick up and move objects around. As they were doing so, I evaluated them in terms of their ability to touch the objects, to grasp the objects, to pick the objects up, and put them down in the right location. And so this is an example of the joint angular velocity control. So the user's finger joint angles are controlling the speed of rotation of each of the robot's joints. So what I found, really surprisingly, was that people could use almost any control mode except for the gripper position and orientation correctly. It was very difficult for them to control that mode. So here on the left, I'm showing the participant's performance of the task. And so the y-axis here is the average stage of that pick up and place task that they're able to complete. If they're up at the top, that means they could complete the task every time. And so you can see in all of the control modes except for gripper position, there's people that can figure out the task and do pretty well. Each one of these dots is an individual subject. And we think that this result is significant for 2 reasons. One is that, although we didn't find a single control mode that's easier to use than the gripper velocity, we've opened the door to new approaches by showing that we need not limit ourselves to gripper velocity. There's other control modes that we can explore that are also viable. And then secondly, it's really fascinating in its own right that people just can't learn to control the gripper position and orientation. They really struggle with that. And so some of our future work is going to try to address the question of why that is. So I just want to take a second to thank the folks that helped make this possible. So Pedro Alcalea, PhD student in the department, huge help, and Angel Labrada Galvan, who is an undergraduate research assistant working with me, who is also a huge help to the project. So thank you both, and thanks to the NSF for funding our work.
Question for Tanat.
Oh, yeah.
So, for the previous slide, when you were showing the difference between the different modes, was there any statistics done on was there statistical significance between the different groups? Because it looks like they kind of go into each other.
Yeah, the only one that's statistically different is the gripper position. Yeah.
Wonderful.
Thanks so much.
All right, thank you. So we'll stick with our Atlanta cohort. And let's talk and get into Pedro Alcalea.
Hello everybody, my name is Pedro. So hello everyone, my name is Pedro, and today I'll be presenting you about improving brain-computer interfaces. So to start off, click on the screen over there. So to start off, I kind of want to introduce what BCIs even are. And in the first place, you can think about them as systems that harness your brain signals and then use these brain signals and try to use them to control something external. So let's say a computer cursor or a prosthetic arm. And these systems are particularly useful for people with paralysis, because otherwise they wouldn't be able to control anything in the external environment as they cannot move any part of their body. So we use these systems to help these individuals control a computer cursor or a prosthetic device externally, correct? However, the control isn't so good, so don't imagine anyone's going to be walking like this anytime soon. Basically, the real control state lies somewhere around here, where even controlling a computer cursor with this BCI can be very difficult. As you can see, the cursor is moving very jittery, it has unreliable stopping, and not so good accuracy. And this all comes from the noise that comes from our brain, as our signals are not uniform per se. And I want y'all to think about this control as if you were using a controller joystick, except you're forced to use a joystick with a chopstick away from it, making the control as hard as you can imagine. And over time, several things have been tried to improve this control, starting off with the Kalman filter in 2002, and later on the linear regression in 2018. And of course, they've tried many other methods in the past in order to slowly try to improve this technology over time. And I want y'all to think about this as the improvements of game control technology over various years in history. But alternatively, we came in and said, what if we try some different type of system that doesn't involve continuous control? What if we decide to constrain commands to forward directions and stopping only? This resulted in some type of control that looks more like this. As you can see, the movement seems to be way more controlled and towards the target than we previously saw in these continuous cursor movements. And for this example, I want y'all to focus as if we are controlling it using the D-pad instead of the joystick now. And basically, we decided to test all these decoders and see which of these actually performs the best. To start off, researchers did actually achieve success in making the linear regression decoder of 2018 far superior than the Kalman filter of 2002, with a 43% success rate. But our discrete decoder DDS kind of blew that out of the water completely. And that brought us down to a question, which was, does discretization alone make this performance so good in the first place? And this brought us to the hypothesis, well, if this is the case, then we believe that DDS will perform identical to a discretized linear decoder, as we saw previously. And we believe that this discretized linear decoder will also perform better than its continuous counterpart. And to run this test, I want you guys to remember, think about the discrete decoder as just a D-pad, as the linear decoder as just a joystick, and then as the discrete linear decoder as if it was a joystick, except now we're forcing the joystick to only do one of 4 commands, or stopping, of course. And we ran this test using this little model we like to call the Job BCI, and the reason we did it is because it allows us to get a total of 108 subjects to run this experiment due to the non-invasiveness of using the brain as a signal input rather than, the hand as a signal input rather than the brain. And essentially we had each subject come in for 100 total minutes and had to perform this task where they had to control the computer cursor to one of these 8 targets at every single moment in time. The more they hit, the higher the success rate. And now, when we ran these groups compared to each other, we saw that the discrete continuous decoders actually improved performance by over 20% success rate, showing that discretization does indeed improve performance at a significant amount. However, DDS, our previously mentioned decoder, performed far superior than all these methods combined, still with 87%, showing performance that's 26% greater than any of the discrete counterparts we created. So this left us with 2 things. We now know that discrete decoders do indeed improve performance of these BCIs, and it seems to be related to the fact that they reduce noise that neurons cause due to their modulation in our brains. However, we still have left to answer the question, what about DDS specifically is what made it so much better than all the other decoders? And I believe it involves something we haven't looked at throughout this whole presentation, and involves the components that cause this discrete decoder to even exist. And that is what my work will focus on in the future, to find out what it is. Thanks for listening, and thanks for all you guys.
Question for Pedro. Go for it. Do you find that increasing the number of possible discrete selections changes performance? Like, do people get worse if you have more selections, do you think?
So the only anecdote I have of this is that now we're actually trying to make a robot controllable with this device, so maybe Dr. Octopus is possible. But the control scheme to do this has been very difficult, and it seems that every additional command you add actually reduces the certainty that the decoder has of you picking that command. So control seems to waver a little bit the more options you give it.
Our last, but certainly not least, presentation is from Pritam Kumar Saha.
Hello everyone, Kumar Saha. I'm graduate student in Dr. Wolf Kishanwar lab. So today I'll talk about neuromodulation and neuroplasticity in sleep-disrupted post-traumatic epileptogenesis. So traumatic brain injury is a global health crisis. If you look at global scale, almost 69 million people's TBI cases globally every year. If we narrow down our focus on USA, we can see that 1.7 to 3.7 million TBI cases in the USA annually. If we put into the perspective, it's a total population of combining Los Angeles and Philadelphia. Among the many outcomes of the TBI, long-term outcomes of the TBI, sleep disruption is one of them. But there are two phases of the sleep, is in REM and REM. On the TBI, both phases are affected. If you look at the statistics, we can see that almost 50% people of TBI injured patients suffer from hypersomnia, 25% suffer from insomnia, and others also suffer from different abnormal behavior. But to find the main causes of that, there are multifactorial mechanism underscore the need for preclinical model to link injury to delayed outcomes. So it is important to find the central regulator that actually causing it, to find a suitable intervention approach regarding these issues. So among the many multifactorial mechanisms, we focus on the GABAergic mechanism because this is one of the central regulator of the sleep, and more specifically, we focus on the PV neuron and GABA uptake. So how GABAergic dysfunction alters sleep and excitatory inhibitory balance is remain unclear. So we hypothesize that chronic sleep deprivation prior to TBI worsen the whole conditions, and TMS treatment probably are one of the probable options for the intervention of this kind of scenario. We pick TMS treatment because it has many benefits. It is non-invasive and cost-effective. It has potential for neurological and psychiatric disorder-related issues. It can curb cortical hyperexcitability. It also has a potential to improve sleep quality. This is our experimental design. We started from the injury to 60 days after the injury, and meanwhile we also perform TMS treatment 14 days continuously after the TBI, and then we check the EEG. We do the EEG circuit, collect EEG data, do the sleep analysis, and also we do the histopathology. And in terms of results, we see remarkable results. We saw that TBI injured patients suffered from sleep fragmentation in their NREM sleep, and we see remarkable recovery with the TMS treatment. We see that TMS treatment restored the patient's sleep fragmentation level into the sham level, which was very remarkable. We also see that TBI patient also have suffer from like hypersomnia during their sleep analysis. And now when we look at the GABAergic signaling pathway, we see that the green one is for the GAT, which is the GABA transporter. We see that the GAT expression reduced in comparison with the sham after the injury, and with the TMS treatment, we see remarkable outcomes. We see that TBI and after TMS treatment, their GAT expression has almost come to the optimum level as previously, and you also see the IBA1, which is microglia activation. It suggests the microglia activation After the injury, IB1 expression was increased. After the TMS treatment, we see that the results also comes down close to the their previous before the injury level. And then when you look at the PV interneurons, which is very important because of its firing and excitability controlling mechanism in GABAergic mechanism. So we see that in sham, in comparison with the TBI, the PV expression and intensity and density both reduced in the injured cases. But with the TMS treatment, we see again a very excellent outcome. We see that the TBI patient shows that recovery in the PV intensity and PV positive cells density. Similarly, when we see the PV results in their dentigerous region along with the CA3 region, we see that in the dentigerous region, we also see the TBI patients with sleep deprived suffered from reduced PV intensity and reduced PV positive density. With TMS treatment, we see the value has been rescued almost to the optimum level. In summary, TBI actually disrupt the GABAergic mechanism and also disrupt the sleep-related all the parameters, and TBI increase the fragmentation. But with the TMS treatment, we see some positive results. We see is that maybe this can be one of the possible intervention approach that we can actually look forward for the future results. That's all. These are the findings, and thank you to all my lab members and Dr. Shamraj.
Question for Pritam? Thank you to all the speakers for staying on time. We finished with 1 minute to spare, so that's pretty amazing for two speakers. So I really appreciate it. We are, so let me reiterate again, our grad students are doing fantastic work, and as I said before, truly believe, not truly believe, it is true that the grad students, especially our PhD students, are the engines of our department, and we really value all the work you do. It's a great job at the poster sessions and with these oral talks. We will move on now to our afternoon special session. I don't know who's introducing it. Right, you want to give it a try? Yeah. So we have a fantastic afternoon session that Dr. Ramella's group has put together, and